Okay, today we will discuss recombinant protein expression. So recombinant, another place where you see this is our DNA. Actually, there's lots of, rDNA can also mean ribosomal DNA, but in most contexts in molecular biology, if you see rDNA, that typically means recombinant DNA. We've talked a lot about recombinant DNA, but maybe not necessarily referred to it as that. So what does the word recombinant mean?
There was recombination.
Yeah, okay, this is an interesting way to say it. I've never heard anybody actually say that before, but that makes sense in some sense, although not necessarily. Like you could produce recombinant DNA through like restriction enzyme cloning. So not necessarily recombination per se. What were you gonna say?
Pretty much that. Like it's like they're combining like different things together.
That's closer? Yeah, like more like recombining different things. So the idea is recombinant, the definition that you should understand is that it's essentially taking DNA from some source, say source A, and then moving it to source or like destination B. So it's essentially taking DNA from one organism, moving it to another. So we've talked a lot about this in the context of like transgenesis or delivery of plasmids often involves recombinant DNA. So anytime you're taking a sequence that's derived from one organism and you're moving it into what's called a heterologous, a different source or a different place, that's recombinant DNA. Okay, so that's what the word recombinant means. So recombinant protein expression then is if you have some gene X that you're very interested in, and typically when you think of recombinant protein expression, you typically are moving it into E. coli for the explicit purposes of expressing protein X. So there's lots of examples where we do this in biotechnology. One of these, for example, is like insulin. So many medicines that are proteins are created in this manner. So things like insulin, you might take the insulin gene, you might clone it into an E. coli expression vector, and you might actually produce insulin that you could give to people as medicine. That's one example. Another example of this would be in the lectures, like if you look back, there's examples where they're taking like spider silk, so genes that encode the spider silk from spiders, and they want to be able to mass produce spider silk to make clothing out of. So one of the first things they tried is moving these genes that encode the major spindlin spider protein into expression plasmids in E. coli. That's another example. So medicines, biotechnological like products. Does anybody have any other examples that we can think of? Biopolymers. A spider silk is actually a biopolymer. Any other examples? But that's essentially like the gist. The gist is, okay, so the gene encodes for a protein, move it into E. coli to get high abundance yields of the protein for downstream usage cases. Okay, so let's talk about first bacterial expression. So there's pros and cons of every expression system. E. coli is kind of like the workhorse for recombinant protein expression for a few reasons. It's fast, it's cheap. These are probably like the main reasons. But there are some downsides in that you don't, if you have a protein that requires, so let's do this is pros, cons. Cons would be that there's no complex folding. So if there was a situation where you were trying to express some key like mammalian protein that required very complicated folding patterns, E. coli might not be the way to go for that, because E. coli, much of protein folding, so if you think of like the primary structure and then the secondary structure and then it forms this global tertiary structure, oftentimes to go from this to this requires other proteins that are called chaperones. So a chaperone is actually an enzyme, a protein enzyme that takes the sequence and folds it and brings proteins to their proper folding, three-dimensional folding. And so E. coli is going to have a totally different set of chaperones than a mammalian set of chaperones. So if you need proper folding, E. coli might not be the best system for that. Another thing that it cannot necessarily do is post-translational modifications. So many proteins, if they're in a eukaryotic context, get made, and then this is not actually the final version of the protein. Often what happens is there's a series of enzymes that come in and make modifications to this thing, adding different groups to this A, and then maybe this is actually the protein that actually like does the functionality. So if you have a more complicated protein that requires post-translational modifications, E. coli might not be the best setup for that. But for the vast majority of like simple proteins, if you can do it in E. coli, that's the best case scenario, because you could just grow vats of E. coli in these giant fermenters, and then you could purify large quantities of almost any protein in particular. Okay, so knowing that, some of the things that are really important for protein expression are the strains. We've talked about a lot of different cases where when you work with organisms in the lab, you're typically not working with wild type. So for example, Drosophila, you're often working with white-eyed mutants. With yeast, you're often working with auxotrophic mutated strains. Cell phone. Sorry, I lost my train of thought. Okay, so there's like lists of special strains that are particularly good at protein expression, and you want to kind of know what these are and be familiar with these. So one of these is called BL21. So this is the strain name. So when it has a strain name, that implies that there's a unique genotype. Okay, and you can actually look these up. So if you Google like E. coli strain database, you'll find a database where you can type in the names of strains, and you'll get a series of listed genotypes. Okay, you'll get a list of things that are unique about that genome. And the one thing that's particularly unique about BL21 is you'll see delta ln. Okay, so delta ln, what's the delta mean? Yes, deletion. So there's a gene in E. coli called ln, and the BL21 has a deletion of ln. So ln is it's called ln protease. It's a protease. Okay, and proteases do what?
Cleave proteins, right?
They what?
Cleave proteins.
They cut proteins. So why would you want to delete a protease that cuts proteins in a strain that's important for recombinant protein expression?
So it doesn't cut what you're trying to.
Exactly. So if you're expressing gene A and you're trying to make protein A, it's not helpful if the E. coli cytoplasm has a protein that's going to come and cleave or destroy your protein. So BL21 is kind of like one of these base strains, which many other strains spawn off of, that has deletion of ln, and there's also deletion of other proteases. So I'm just going to say other things. But the general gist is that BL21 has deletion of some proteases, which enhance the yield of your protein expression. Okay. Then there is BL21AI. So I'm not giving you a complete list, right? Like there's many more. I'm just giving you some highlights so you get a sense of the different genotypes that are available. So BL21AI is a spawn off of BL21. So it's got its deletion of ln, it's got its deletion of other proteases, let's say protease X, and it's got another mutation or unique thing about its genotype. You'll usually see something like delta Arabad. Does anybody remember what this thing is or comes from?
Arabinose metabolism.
Yes, this is arabinose metabolism. So remember, there is the arabinose operon, which is often included on the E. coli chromosome. And if you see, you'll see some variation of this. I think there's probably actually an insert of something else where they inserted a gene to break this stuff. But if you have deletion of Arabad, that essentially means that the arabinose operon, the endogenous one, has been deleted or broken. Why would this be an important deletion? This connects directly back to the plasmid selection.
That way it'll want to take up the plasmid that you insert.
You're saying they would want to take up the plasmid? Well, having the arabinose operon will not prevent them from taking up another plasmid that has an arabinose operon. So let's actually just review, what's the plasmid that actually has the arabinose operon?
PBAD?
Yeah, so there's this PBAD plasmid, which is quite popular. It's got a promoter, it's the ERA promoter, which remember you add arabinose and it turns it on. Okay, and try again to guess or make a connection of why you would want to work with this plasmid in a strain that has a deletion of Arabad.
That way you know for sure that your transformation is successful.
Yeah.
Well, the reason you know it's successful is because it has an AMP cassette. So all of a sudden the bacteria that acquired this plasmid will become ampicillin resistant. That's the only reason you know that it has a plasmid.
So you can control expression.
Like, is it more
Yes, so connect that further? Be more specific.
So if you had the Arabad, like already in the chromosome, it would already be producing arabinose. Like it would digest the arabinose.
Yes, that's the key thing, is if you have this wild type copy and you add arabinose to this media, the bacteria is just going to eat the arabinose, and so your gene might turn on temporarily, but then it will just eventually shut off. So you should start to realize that it's not just a matter of picking the correct plasmid to clone your gene X into. It's a matter of picking the correct plasmid and then pairing with the correct strain. Otherwise, your expression will not work at all. Okay? So BL21AI is a strain that's compatible with this plasmid. And this is true for many different plasmids. Also true for PETs, where with the PET plasmids, which is a different system that relies on IPTG, there are other strains that only that is compatible with in terms of expression. Okay? So you need to understand to be able to look up the different strains, see how they're unique, and see how that compatibility mixes or matches with different plasmids. Okay? So that's BL21AI. Okay, another one of these is called Rosetta. Okay, Rosetta cells. Rosetta cells are also BL21 derivatives. So they're the base genotype of BL21 modified a little bit further. So they also have the delta lon. They're not the AI, so they would have functional arabinose operon. The thing that the Rosetta cells have unique is they have a plasmid called P rare. So P rare is this plasmid with a chloramphenyl cassette. And on the P rare plasmid, there's a constitutive promoter that is driving expression of rare tRNAs. So can anybody remember, or let's say tRNA 1, 2, 3, etc. I can't remember exactly how many there are on the P rare plasmid. I think there's something like 10 to 20 rare tRNAs. Maybe that's too many. Somebody should fact check that. But it's a plasmid of rare tRNAs. What's a rare tRNA, and how can you connect that back to what we previously talked about in the older lectures? What's tRNA do?
Brings the amino acid to the mRNA.
Yes, tRNA is like this little RNA that binds to a given amino acid that reflects the three essentially like digits that it's binding to. And the 3 digits, another word for that is what?
Codon.
The codon, okay? So there's some amino acid, let's say this is alanine, which has a tRNA that recognizes it, and that tRNA is compatible with a matching codon, so that when the ribosome is building these things together, it's essentially the tRNAs keep funneling amino acids to the ribosome to complete the string. Okay, and so what are rare tRNAs then?
Ones that recognize rare codons.
Yes, exactly. Okay, so now you guys understand. You've made the connection. So remember that all different organisms have different codon preferences, and E. coli is no different. E. coli has its own codon preferences, and it has its own rare tRNAs that are just not abundantly expressed because the native E. coli genome does not have a lot of genes that are utilizing those rare tRNAs. Okay, so the reason that this becomes a problem is in recombinant protein expression, you're taking a gene from somewhere else let's say you're trying to express a Drosophila melanogaster gene Z, and if you're putting that on a plasmid for E. coli, the gene Z is coded for, in theory, in the codon preference of Drosophila, which probably does not match E. coli. So how would this help then is this plasmid is coding for all the rare RNAs that you might find coding this actual gene. So that should, in theory, boost the yield. You should get an increase in the yield of this gene Z if you're expressing it in E. coli. Without this plasmid, you will not get a high yield because the cell is constantly searching for these rare codons, okay? So we all should understand that these are now 3 examples of unique strains with unique strain names that have unique genotypes that do different things and give them different compatibilities for recombinant protein expression.
What does the BL stand for?
I don't know, you'd have to Google that. I don't know.
Because I know the Rosetta is like, the plasmid is the Rosetta Stone, right?
That's probably why they named it. Yeah, that's a clever, I don't actually know if that's true. That makes sense, that the Rosetta Stone is translating, and so that would connect the tRNAs to translating rare codons. Yeah, that makes sense. But I don't know what the BL21 comes from. Probably some boring methodology, like this was BL version 21, I don't know. Okay, let's see. Okay, let's talk about inducing expression. Okay, so the most common ways to induce expression, we've already talked quite a bit about in the context of the operons. One of these is with IPTG. That's typically compatible with the PET plasmids, which if we have time, I'll draw out some PET stuff. Also, there's arabinose. That's typically compatible with the PBAD. And there's typically others, like there's others, but in my experience, these are the most common, the most frequently used. PETs are very common, extremely popular, and the PBAD is particularly popular in my lab and in other labs that I work with. So these are common ones. When you're inducing E. coli, so induction, like the protocol of how you induce expression is actually quite important. So if you are to draw a graph, if we've had some microbiology experience, if the y-axis is number of cells and the x-axis is time, okay? So we're measuring growth over time of E. coli. The growth curve looks like this, okay? And so there's different phases of this growth curve. This first phase is called the lag phase. So this is the phase at which E. coli is, throughout this time, E. coli is pretty much doubling every 30 minutes, except up here. But in the early stages, it's not reaching a point at which it becomes exponential. Okay, so that's the lag phase. The second phase is then the, correct, very good, exponential phase. Okay, and then the third phase, stationary phase, good. That's essentially where, what's happened at stationary phase?
It's just like produced all it can, and I think it's like eaten up everything it can.
Yeah, this is the point at which the resources, the food, runs out. And it just can't, like nothing can, you've heard that, well, if bacteria double every whatever, eventually you cover the whole planet.
Yeah.
The reason that they don't cover the whole planet is because there's a limited amount of food. So when the food runs out in the media, that hits stationary phase and they stop reproducing. And then some people will say there's like another phase, like death phase, where they eventually start dying. But I don't know how far out like that is. Like bacteria can survive for a long time.
It was pretty long, and then it like dips down.
Yeah, but bacteria can survive for a very long time. So for recombinant protein expression, essentially like these phases are the most important phases. So then the next question is, which of these phases is the best to add your inducing agent? So these would be called the inducer, IPTG or arabinose. That's what's actually turning on the protein. When would be the best time to add the inducer?
Wouldn't it be the exponential phase?
So I heard lag phase and I heard exponential phase.
Log phase.
Log, is that's what you mean by exponential phase? So you're saying, okay, so yes. So this is, the exponential phase is the best phase at which to add the inducer. You don't want to, why do you not want to add it in the lag phase?
It's not really doing anything at the moment.
Well, as soon as you add the inducer, the entire metabolism of the organism will shift. Because if you have, imagine you have like a 600 copy plasmid in a cell, and you give that cell arabinose, that essentially means that the entire metabolism of that bacteria will shift towards creating protein X, whatever is in there. So if you give the inducer a lag phase, it shifts metabolism so that you will not get good reproduction. So you'll never get a good yield because you don't have enough cells. So it's gonna say, essentially if you add it too early, you're creating a fight between reproduction and producing your protein. So you want them to be able to reproduce enough. So ditto, why would you not add in stationary phase?
They've used up all the resources.
They've used up all the resources, so they now have no longer energy to produce, so you're gonna get a low yield. So you get a low yield here and you get a low yield here, and your goal is to produce the highest yield. So actually the best time to add the inducer is right in the middle of exponential phase so that they have grown enough, but yet there's enough food left to switch metabolism to producing the protein. Okay, so this point, you can measure this. So then the third question would be, how do you measure in E. coli culture simply the number of cells? Does anybody know?
Oh, I forgot
Serial
Yeah.
distillations.
What?
Serial distillations.
You can do serial, so you can do serial dilutions.
Dilutions,
You would have to do dilutions before you run the analysis. That's very common.
Optical density.
Yes, it's the OD. So the term here is OD, which stands for optical density. How do you measure optical density?
590 nanometers.
So that's, I think it's usually 600 nanometers.
Spectrophotometer.
Yes, that's what I'm looking for. So the instrument, the quote unquote instrument that you would use is called the spectrophotometer or the spec. So you can use a spec, you set the wavelength, the lambda, which is I think it's the absorbance. Hopefully, who has used a spectrophotometer? A few people. Usually this is like, you usually use one of these as like a standard lab in chemistry. Hopefully they'll do that more. It's pretty simple machine. You just turn it on, you change the wavelength. So you're changing the wavelength, and what's happening with the spectrophotometer is it's essentially, if I was to draw a spectrophotometer, it looks something like this, where there's kind of like an opening part, okay? And you have what's called a cuvette, and you fill your cuvette with your your media that has your cells in it, okay? So you have one that has cells in it, and you compare that to, hopefully people can still see this, you compare that to a negative control. What would your negative control be?
LB media.
Yeah, it's just LB, so it's just empty media that has nothing growing in it. You would put your LB in there, and you press the blank button, which essentially takes a baseline reading of the LB. Then you switch the cuvette, you put the cuvette that has your cells in there, and then you press the read button, and it'll spit out a number based on the cloudiness of the media. And the cloudiness of the media for E. coli is an approximation of the number of cells.
Oh, wait a
So,
second. I think I might actually have used these with like fish, like doing, what do you call it, like testing magnesium levels or whatever.
maybe. So a spectrophotometer is a very common instrument. This is not the only thing it's used for. It's essentially like a drill or a hammer to a mechanic.
Yeah.
The spectrophotometer is to the scientist, if that makes sense. Like you can use it in a ton of different stuff. So we have one in our lab. This is most commonly what we're using it for, to just get a quick readout on the number of cells. So when the OD 600 nanometers reaches 0.5, so it'll spit out a number, which is 0.5. That's when you've reached the mid of exponential phase. So it's kind of like this magic number, OD 600, 0.5 reading. That's when you want to add the inducer. Okay, so this is a key, crucial thing. If you ever do recombinant protein expression, please remember this. I worked with people who didn't understand this, and they wasted years of their PhD trying to induce proteins at the wrong time, and lost years of their life because of this. So this is a very important thing, and it's also just kind of like fundamental microbiology for recombinant protein expression. Okay, so that's when to induce. How much time do we have left?
25 minutes.
25 minutes, okay. debating what to do next. Okay, so just couple other kind of like quick things for recombinant protein expression. So we've all have experience from our lab with one type of media, which is LB. There's other types of medias, okay? So just know that there's other recipes, and at some point you might want to switch to other medias depending on your yield. So if you try to express a protein in your LB and you're not getting a very high yield, you can sometimes fix that by switching to other medias. One's called terrific broth, TB. And essentially, like, there's just a series of other medias that are adding different nutritional nutrients for the E. coli. And by bolstering the nutrients, sometimes you can reach a higher optical density, which will give you a higher yield of protein per cell. So sometimes you might have to tweak the media. Okay, let's talk about shakers. So when you grow E. coli, usually you have what's called an Erlenmeyer flask. Erlenmeyer flasks look like this. The rule for filling the Erlenmeyer flask, how full should the Erlenmeyer flask be? Does anybody know? If you're doing recombinant protein expression. So you fill this with what? Media, so you have some media in here. How full should you make it?
About right there.
Yeah, halfway. Okay, good. So you fill it halfway. Why do you only fill it halfway? Was that just a guess, a good guess?
To leave headspace. Right?
Why do you leave headspace?
Well, I don't know.
Okay, so this is good then, because I'll actually explain. So, okay, there's multiple reasons, I guess. One, this is gonna shake. Okay, so because it's gonna shake, like, you don't want it spilling everywhere. That you don't want that. That's bad. Two, E. coli like oxygen. Okay, E. coli essentially like need to breathe. So in order to get oxygen distributed into the media, that's why you shake it, and that's why you leave it half empty, is so that there's enough air in here that when it shakes, you can get good aeration. So you need oxygen. Okay, so that's shaking. Typically you have a shaker, so you have like a plate that holds this thing, and it'll just go like this. Okay, so it'll shake it, and usually you are growing these things at 37 degrees Celsius, which is the temperature that E. coli likes. And then when you add the inducer, you can do two different things or multiple things. Some people will change the temperature that the expression happens. So if the first part is growth, so essentially growth to OD 0.5, 600 nanometers. In that phase, oftentimes you're just growing at what E. coli likes, 37. But then after you add the inducer, sometimes people will shift to like 18 degrees Celsius, so like a cold expression overnight. So they'll just let the E. coli shake overnight at a cold temperature, and you're slowing down the growth. And sometimes that can optimize the expression of a particular protein, and some particular proteins might like to be assembled when it's colder, depending on what the protein is. Other proteins are fine to just do 37 degrees Celsius for 4 hours. So these are kind of two standard things. You either keep it the same and express for 4 hours and then you harvest, or you downshift the temperature and express overnight and then harvest in the morning. Sometimes, depending on your protocol, like one of these might be good depending on the particular protein. Every protein is different, so you're gonna get different results based on the uniqueness of your protein. Okay, so once you've grown it, you've shaken it, you've added your inducer, you've now done one of these things, you've now got a situation where you have a flask full of cells that have your protein inside of the cells, okay? So then the next step is, how do you recover the protein? What has to be done next? Yes, lysis. So the next step would be lysis. I don't know what step we're on. Lysis. Okay, so lysis in E. coli. E. coli has a cell wall, so that can actually be kind of difficult, okay? So there's different methodologies for lysis. Does anybody know any?
You have in the notes
Adding
a
it
French
to
press.
Let's talk about that one. That's one of them, definitely. Let's list them out. So there's often you see freeze, thaw. That's kind of like simple, easy one, but not super efficient. So sometimes people will take the cells, they'll flash freeze them, and then they'll thaw them. And when you flash freeze cells without glycerol, the ice crystals will pop open the cell and you can break the cell wall. So some people will freeze thaw. Oftentimes this is just convenient too, because maybe expression takes a whole day and you want to go home. So you flash freeze the cells and then they're kind of like set until you come back ready to like recover. So oftentimes this is a part of the protocol. There's another, what's called enzymatic lysis, which uses an enzyme called lysozyme. Hopefully I spell it right, lysozyme. Lysozyme comes from chicken eggs. So chicken eggs have an inherent enzyme called lysozyme, which dissolves the cell wall of tough bacteria. Why would chicken eggs have an enzyme that dissolves the cell wall of bacteria?
Because they need to hatch from the egg.
Yes, they need to protect themselves. So it's essentially like an immunity protein that's expressed highly abundantly in eggs. So you can purify that from eggs, and it's a common molecular biology reagent. Usually every biochemistry lab has lysozyme in the freezer. So you'll just take what's called a pinch of lysozyme, you'll open up the container, you'll grab a pinch, and you'll just kind of like drop it in your Erlenmeyer flask, you'll shake it around, and that just kind of like enzymatically will dissolve the cell wall. And then there's often what's called a sonicator. A sonicator usually looks something like this, where you have like a probe, you'll take a tube with some cells pelleted in it. So oftentimes before you do any of this, you'll take this, you'll centrifuge it down, collect the cells, get rid of the supernatant, which is the media. You'll put your cells in a smaller volume, and then you can take that tube and you can hold it in the sonicator, you press a button, and then the sonicator releases ultrasonic waves, which can destroy the cells. That's a pretty common, easy way to do it. And then another way, it's becoming less common. We used to have one, although now it's broken, the French press. So the French press is this kind of like machine, not a coffee French press, but it's a machine that has a cylinder like this with a plunger. There's actually a video on this in the playlist if you want to see this. And you can fill the inside of this. There's a cavity, okay? And then there's an exit port, which usually you port into a separate tube, okay? And you put your cells in here, and then you put the French press in a giant machine that squeezes it down and squeezes it together, and then the pressure in the chamber becomes super high, and then as soon as you open up this hole, it pops all the cells, and then they slide out through the container. So these are various mechanisms of lysis. There's multiple ways to do this, there's more, but these are some very, very common ways. Essentially the point is you have to break open the cell to recover the protein, okay? So once you have done lysis, what's the next step?
Purification.
Yes. So then the next step is purification. Become pure. Okay, so once you lyse the cells, you have this, essentially you're gonna have some situation where you have some tube, okay? And you've lysed, and so you have released all the cellular contents into some buffer, okay? And then usually what people will do is they'll centrifuge, they'll get what's called the pellet, and they'll get the supernatant. And so there's a question of which of these fractions, after lysis, after you centrifuge, which of these fractions probably has your protein, and why?
It's pellet, but I don't know exactly why.
You think it's pellet? Does anybody think it's soup?
I think it's soup.
Why do you think it's soup?
Because the rest, it's like the heavy cell wall stuff fell to the bottom.
Yeah, so often, so this is a complex question, and it matters, it's context dependent depending on the protein. So you're correct that what's usually in the pellet is kind of like large cellular chunks. So like cell wall stuff, and other things that are quote unquote insoluble. Okay, so insoluble stuff goes into the pellet, and soluble stuff, which means it's dissolved, it can dissolve in the buffer, which is usually a baseline of water. So soluble stuff will stay in the soup. Okay, so if you're expressing, let's think about this. So if we have gene X and we have gene Y, and let's say gene X is found localization in cytoplasm, which fraction would probably contain protein X? Soluble or insoluble? if the protein is naturally localized in the cytoplasm?
Soluble.
Soluble, because cytoplasm is essentially water. So any protein that is in the cytoplasm, free diffusing in the cytoplasm, is by its nature soluble. So you'll find those types of proteins in the soup. Most proteins are soluble, okay? There's many other proteins that are insoluble. So for example, membrane proteins. Membrane proteins are, by evolution, evolved to be comfortable in lipids, and lipids are not soluble in water, okay? So if you're trying to purify a membrane protein, your protein would probably be mostly in the pellet, okay? So keep in mind that at this point, your protein could either be here or here. It could be a mixture. It could be 90% in soup, 10% in the pellet, depending on what the protein is, the amino acids that code it, and what it does and where it localizes to, okay? So that's often a big thing. Proteins that are insoluble are often pretty difficult to work with. Proteins that are in the supernatant are usually much more friendly, okay? So, okay, now we have to get to purification. So the first part of purification is figuring out, is it here or is it here? That's usually the first part. So you can usually figure this out by running, what kind of a gel would we run? What would this be called?
Are we doing the protein?
Yeah.
SDS page.
So you could just run a simple SDS page, and you would take a little bit of the pellet, dissolve it in the buffer. So you put maybe pellet here, and then you take a little bit of the soup, and you could run it here. And if you saw, you say you had a ladder, if you saw a pattern that looked like this, then you would know that your protein was soluble. If you saw a pattern that looked like this, you'd say it's mostly soluble, but there's some in the pellet. If you saw a pattern that looked like this, you'd say, okay, it's insoluble. And so this is a quick experiment you can do real quickly to figure out, after this, there's gonna be a different pipeline of purification depending on like where your protein is, okay? So given that it's soluble, then usually what happens is some form of chromatography. And we've talked a lot about a very specific form of chromatography that involves tags. So that's affinity chromatography. So usually you'll have, if you're doing recombinant protein expression, you'll usually in your plasmid have something like a his tag. Talked a lot about that in the past. That will have 6 histidines. That will allow you to run that protein in the supernatant. So you take the supernatant and you would run it over a column that has cobalt beads, or you can sometimes use nickel beads. You'd run that over the column, and then your protein will stick to the beads while everything else will drip off, and then you can just filter or run buffer over this to do what's called wash, to wash your protein. And every time you run more buffer over, your protein is still sticking to the beads, but every contaminant is being washed away. So your protein will get more and more and more pure based on washing it more. Okay, and then the final step, so if one step was affinity purification, then the final step is called what?
Elution?
Yes, elution, very good. So the final step of this is typically elution, where you add a reagent. So now if your protein is essentially saturated, bound to the beads, you have to get it off and recover it. So after you've done your washes, you will strategically then put a tube underneath to catch the drips, and you will add the elution buffer. If it's a cobalt His6, the elution buffer includes imidazole, which you should probably be familiar with this, very important biochemical reagent. Imidazole outcompetes the His tag for the cobalt binding. So when you add imidazole, it's a tighter binding to the cobalt, and it will disperse the protein away from the beads. And so then what will happen is your protein will drip off into the tube, and you've caught it now in the tube. So now you have essentially a very, very highly pure protein X that you purified in your tube. Okay, final step of recombinant protein expression, after elution. After elution, what needs to happen? What do you need to do in this? Are you just satisfied with this? It's got your protein, yes, but what do you want to know about it? You want to know how pure it is, and you also want to know the yield. So the next, usually the last step is essentially some kind of a yield calculation. How much time do we have left?
10 minutes.
Okay, good. Okay, okay. So the next phase, the final phase, usually what people will do is if you've spent a lot of time purifying protein, that's a very labor intensive thing. And often that protein is very valuable to you because you might have spent like a whole week focused entirely on purifying this protein. So oftentimes what people will do is they will aliquot this. So they will set this up and they'll distribute the distribution into multiple tubes. You'll add glycerol often to many of the tubes, and then they'll flash freeze it, put it in the freezer. So proteins are usually, most of the time, pretty stable. If you add a little bit of glycerol and you freeze them, they can be pretty stable even if they're enzymes. Okay, and then usually you'll take one of these samples and you'll do some kind of a yield calculation. So there's different ways to measure yields. Okay, one of these is called a, we should have room over here. One of these most common ones is called a Bradford assay. If you ever do any biochemistry with proteins, you'll eventually have to do a Bradford assay. Okay, so a Bradford assay is essentially, the very simple gist of it is you add a reagent that binds proteins and makes them blue. And then the amount of blue is a direct readout of how much protein you have. Okay, so then the next step is then you measure the amount of blue. How do you think you measure the amount of blue? Yep, okay, so now our friend, the spectrophotometer, again, you just set the wavelength to read blue color, because blue is right in the light spectrum, it's a set wavelength. So you can calibrate your spectrometer to read how much blue is in there. Okay, so then you could see how you would easily just have a little bit of your sample in that cuvette, and you'd get some number. Let's say you get some number x. Okay. The final question is, how do you know? So let's say the spectrophotometer spits out a number that is telling you how much blue. How can you actually calculate a molarity of the protein based off this amount of blue? Does anybody have any idea?
Also like an SDS page to determine size, maybe?
So oftentimes people will do an SDS page.
Can you make some kind of curve?
Yes. Yes. Okay. So let me go back to the SDS page in a second. That's what you need to do. You need to create so you would compare against a standard curve. Okay, and the way that you construct a standard curve is imagine you have 4 tubes. And in this tube, you purchase some boilerplate protein. The boilerplate protein that people use is called BSA. BSA stands for bovine serum albumin. This is another, you want to memorize this, you want to learn this. It's a very, very common molecular biology or biochemistry reagent. It's essentially just like if you want any old protein, you can use BSA. Albumin is the most abundant protein in blood, so it's very easy to purify. You just collect cow blood and you can purify the most abundant protein, which is albumin. So BSA is very cheap, you can buy it. So imagine in these tubes, you put something like 0.25 milligrams BSA. And here you put 0.50 milligrams BSA, 0.75 milligrams BSA, and 1 milligram BSA in each of these tubes, okay? And then you add your Bradford reagent, which makes, because this one has 0.25 mgs less protein, so there's less protein over here, more protein over here. So maybe this one is a little bit blue, maybe this one is pretty blue, maybe this one is much more blue, and maybe this one is deepest of blue. That's what it would look like in the tube. And then you do your readings. So you do your spectrophotometry readings of these different tubes. Then you could plot that. You would plot on the y-axis the blue, and you would produce a line of these 4 points that is a standard curve that is directly correlating the milligrams. So on the x-axis is the mgs, milligrams of protein. It's directly correlating the milligrams of protein with the blue color on the y-axis. And then all you need to do is plot this x number. Wherever this x number fell, you can use this to then y equals mx plus b. It's a straight line, so you can just use the line formula to calculate the exact milligrams of your protein, and from the exact milligrams of your protein, you can derive a molarity with a calculation based on the amino acid sequence. Okay, so this is a frequent way to do a yield calculation. I'll just do one more example real quick. This is not the only way to do it. You can actually use an SDS page, which I think you suggested. That methodology is called densitometry. You do the same thing where you create a standard curve of BSA, except you run the standard curve on the SDS page gel. So in this case, you have increasing BSA, so you'd have something that looked like this. Okay, and then you would run your sample. Let's say your sample gave you something like this. Your unknown, okay? And then you can produce another standard curve, except on this axis, it's still, on the x-axis, it's still mgs, milligrams, but on the y-axis, it's essentially like darkness of the band. So you can actually have computer programs tell you how many black pixels, like quantify the black pixels, here, 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 okay? That will produce your line, and then you can do the same thing with your unknown, and you'll plot that, and use the y equals mx plus b to again calculate. So these are two ways you can derive the yield calculation. That will tell you exactly how much yield you get of your protein. Okay, do we have any time left?
Yes.
How much time?
3 minutes.
3 minutes. I don't know if that's enough to do complicated stuff.
Could you explain nanodrop?
Nanodrop, okay, yeah, okay, that's another yield calculation. Nanodrop, so there's pros and cons to each of these yield calculations. Nanodrop is kind of considered like sloppy, but like decent. So nanodrop is essentially just you have, it kind of looks like this, and there's like a thing like this, like a pedestal, and it's connected to a computer. And you'll take a drop of your unknown yield protein, you'll put the drop on this thing, you'll close it, you'll just press a button, and it'll spit out a series of numbers that are measuring. It's essentially a mini spectrophotometer that takes a bunch of readings, and it'll give you a bunch of readings. And the reason that you can do this is physically, different amino acids absorb different wavelengths, and certain amino acids can be used to create a standard concentration of how much protein is there. So you'll get some numbers based on different absorbances, and you can use those numbers to calculate a yield. The downside of the nanodrop is it's sloppy. For proteins, it's sloppy. Nanodrop is more useful for DNA quantification through the same methodology. It's a little bit sloppier for proteins. So I'd say that this one is kind of like fast and sloppy. In my experience, in my hands, the best, most accurate way is actually densitometry. But because this involves running a gel, many people will often do the Bradford. So each of these has pros and cons. I think this one is actually the most accurate. But you can do multiples, or sometimes people will even do mixtures. Okay, we can end there. That's the gist of recombinant protein expression. Next time we will have a paper that will discuss recombinant protein expression of Cas9, which connects to the CRISPR stuff that we talked about in the past.